النباتات في التنمية الزراعية المستدامة، وهنبتدي النهارده إن شاء الله بالبروفيسور. وهنبتدي بالدكتور بروفيسور دكتور واي وينج من الصين، محاضرة بعنوان الحشرات الناقلة للفيروسات.
Please, Doctor Y Wing, open your mic. Dr. XU Y Wing, please open your mic.
Dr. Ashutosh, are you here?
Post. Let it be on. You keep yourself mute. Mute you keep no.
Please open your mic, Dr. Ashtut. Please open your mic. Dr. Ashtut, please open your mic.
فصل معاك، فصل النت من عنده.
هو ظاهر معايا.
ما انا كلمت واحد ما دخلش.
You hear me?
هو مش connecting. Yes.
أنا ظاهر عندي موجودة، ظاهر عندي. خرج تاني.
Dr. Wai Weng, are you here? ما هو مش هنعدي 2 ده التالت والرابع.
Dr. Wai Weng, you can hear me. You can start. Dr. Wai Weng.
So, are you talking to me?
Yes, yes. You can start now.
It's my time. I think it's 8 for me, right?
Because the first one have some troubles in net. You can start
Okay,
now.
so I can start earlier, right?
Okay.
Okay.
Please wait to Dr. Noah to introduce you.
So I share my screen.
Yes. Dr. Noah, you can introduce Mr. Wai Weng.
بروفيسور دكتور واي وينج من دولة الصين هيكلمنا النهارده عن الحشرات الناقلة للفيروسات. You can start دكتور واي.
Okay.
Okay. So hello everyone. Thank you for inviting me to join this symposium. Okay, it's my great pleasure to talk with you. The topic of my presentation is whitefly transmission of plant viruses. And I'm Xiaowei Wang from Zhejiang University, China. So I just want to introduce so where I'm from. I'm from Hangzhou. It's a city in East China. It's very close to Shanghai. Actually, Hangzhou is a major tourist city. It's very famous for its lake and for its forest. This is the famous lake, West Lake. And I'm from the Zhejiang University. So it's a top university in China, and it has more than 30 colleges with arts, science, medicine, engineering, and social science. For me, I come from the College of Agriculture and Biotechnology from the Institute of Insect Science. Actually, there are several Egyptian students and postdocs now study or working in our institute. So my major research interest are insect transmission of plant viruses and insect-plant interaction. So today, I will mainly talk about insect transmission of plant viruses. So first, let's look at the plant viruses. How to remove this? So there are more than, the plant viruses are abundant and widespread on the earth. So far, more than 1,200 plant viruses has been identified. Of course, there are many more waiting to be identified. And actually, all crops are susceptible to virus infection. Plant virus has caused huge yield or quality loss to agricultural production, such as cereals, fibers, vegetables, and fruit. And worldwide, the damage caused by plant virus is about $60 billion per year. So the vectors are very important for plant virus transmission. How to remove this? More than 85% of plant viruses are transmitted by vectors. However, only a small fraction of animal viruses are transmitted by vectors. This is mainly because for animal viruses, they can be disseminated by host self-motility. For example, if people got infected by COVID-19, if he go to another place, this can transmit the virus. But for plant, they are rooted and lack independent mobility. Therefore, it must dependent on vector to transmit the plant viruses. And plants also have a thick epidermis, so it can preventing the entry of virus. The insect is the most important vectors for plant viruses. And among the 1,200 plant viruses, more than 75% are transmitted by insect vectors. So when insect vectors feeding on an infected plant, it can acquire the virus. Then when it flies to a healthy plant, it can transmit the virus. That's why the insects are very, very important for plant virus spread and transmission. So this slide shows the plant viruses transmitted by vectors. So on this side, it shows the family name of the plant virus. This is the genus name of the plant virus. And in the middle, it shows the vectors for those different plant viruses. And each color shows different vectors. For example, leaf hoppers, tree hoppers, aphid, whiteflies, and plant hopper. You can see from this slide, most of the important vectors are phloem-feeding insects. And for example, aphid is a very efficient plant virus vector. It can transmit more than 15 families of plant viruses. Another important vector is whitefly. It can transmit more than 400 plant viruses in 5 different families. And in the last 10 years, I have been working on whitefly transmission of plant viruses. So let's take a look at the whitefly. Whitefly is a global pest. Also, it's very tiny. It's only 1 milliliter in size. But when it outbreaks, it can cause huge damage to agricultural production. This picture shows the whitefly outbreak. I just want to mention that the butterfly Burmese tabaci is not a single species. It's a species complex. It's including at least 36 different captive species. And among those species, the bee and fly and the cuvette fly are the most important. During the last 20 years, the bee and the cuvette fly have invaded more than 100 countries around the world, including Egypt. And those bee and cuvette flies have replaced the native butterfly species, and they are the top 100 invasive species. The bee and the cuvette fly also invaded China since 1980s. So now it has spread all over China. So in the South China, it's the bee butterfly is dominant. In Central China, cuvette fly is dominant, and in the North China, both bee and the cuvette fly exist. So except direct feeding, the butterfly cause damage mainly by transmit bigoma virus. So the bigoma virus is a plant single strand. It is transmitted exclusively by the butterfly in a persistent circulative way, and with the spread of butterfly all over the world, bigoma virus has invaded more than 50 countries around the world. It has caused great damage to tobacco, to cassava, to cotton, which I know is important in Egypt, the cotton, and to tobacco. And also, so far, more than 400 different bigoma virus has been identified. Oh, I hate this. And different viruses are transmitted by different Burmese tabaci species. So now let's take a look at the route of bigoma virus transmission. So when the butterfly feeding on an infected plant, it can acquire the virus. The virus can go to the butterfly midgut with the food, and some virus can cross the midgut barrier and into the salivary gland. And then it can be secreted when the butterfly feeding on a healthy plant. Therefore, it can transmit the virus to a new plant. So all these things happens in a butterfly which is only 1 milliliter. So the detail of virus transmission is largely unknown. And during the last 10 years, I have been trying to answer the following two major scientific questions. The first is how bigoma virus cross the cellular barriers of butterfly to transmit. Secondly, both butterfly and virus need to survive on the plant. So what's the plant-mediated interaction between butterfly vector and the bigoma viruses? So the first question is how bigoma virus cross the cellular barrier to be transmitted. It cannot move through cell membranes freely. So how it can transmit across the midgut, salivary gland, and airway barriers to be transmitted. And previously, several studies had shown how it transmits the midgut. So today, I will try to answer how the virus across the cellular gland and airway barriers. So in our lab, we have two butterflies, bee butterfly and a Q butterfly. We also have two viruses. One is TYLCV, tomato yellow leaf curl virus, Another one is tomato yellow leaf curl China virus, TYR-CCNV. Interestingly, for bee whitefly, it can transmit those two viruses very effectively. However, for Q whitefly, it can only transmit TYR-CV, but cannot transmit another virus. So this is probably due to the fact that Q whitefly cannot acquire TYR-CCNV. So we tested this possibility. We put whiteflies on TYR-CCNV infected tomato plant, and at different days we collected whiteflies and measured the amount of virus in whitefly by quantitative PCR. So our result shows actually both bee and Q whitefly can acquire this virus, and actually the Q whitefly can acquire and retain more TYR-CCNV. So it suggests that the reason Q cannot transmit this virus is probably due to it cannot pass some cellular membranes. So we dissected whitefly, dissected midgut, hemolymph, cellular gland, and the saliva, and detected the presence of TYR-CCNV in the organs of whitefly. We can find for bee whitefly, the viruses can be found in midgut, in hemolymph, in cellular gland, and also in saliva. But for Q whiteflies, it can be detected in other tissues, but cannot be detected in the saliva. This suggests this TYR-CCNV cannot or very poorly across the cellular gland barriers. We also did various localization studies using the fluorescence in situ hybridization. We can find here both TYR-CV and TYR-CCNV can invade the midgut of both bee and Q whiteflies, and also the cellular gland of the bee whiteflies. Interestingly, only the TYR-CV can be found in the Q cellular gland, but not TYR-CCNV. So it suggests this virus cannot get into the cellular gland, so it cannot be transmitted. Because this TYR-CV genome is very simple, it only includes 6 genes. And the code protein, this gene is the only structure protein, and it can determine the virus transmission. So we compared the CP sequence between the two viruses, and we found there are some regions that are quite different. So we exchanged the code protein of TYR-CV and TYR-CCNV and generated 2 mutant viruses. We found the mutant viruses both can be acquired by bee and Q whiteflies. However, similar as the previous result, the bee whitefly can transmit both mutated viruses, but the Q whitefly can only transmit mutant TYR-CCNV, but cannot transmit mutant TYR-CV. We also checked the localization of the mutant viruses. We found indeed the exchange of the code protein changed the invasion of the mutant virus into the cellular gland. So it suggests the viral code protein and the primary cellular gland determines virus transmission specificity. We also look at the dynamics of these two viruses. They found when Trcv infected cellular gland is first can be detected at 4 hours. Then it's increased and slowly move to the central region of the cellular gland, from where it can be secreted in the saliva. However, for another virus, Trcv, it can never go to the central region of the primary cellular gland. So those results suggest the Trcv cannot pass the primary cellular gland escape barriers of the cuvette fly, and the interaction of viral coat protein with PSGs determines virus transmission specificity, and the secretory region of primary cellular gland determines virus transmission. So next, we look at the trans-airway transmission for big viruses, except as I just mentioned, the whitefly can transmit viruses from infected plant to healthy plant. This is called horizontal transmission. In addition, some big virus can be transmitted from mother to eggs. This is called vertical transmission. And this kind of vertical transmission is also important in virus epidemics. However, whether Trcv can be trans-airway transmitted to the next generation is in great controversy. So some study shows this Trcv can be transmitted from mother whitefly to the eggs and to the nymphs and to the next generation adult very effectively. But later on, some other people shows Trcv cannot be trans-airway transmitted from mother to eggs. And some other study shows this Trcv can be vertically transmitted to the next generation, but it cannot infect a new plant. So we just want to know why different studies obtained different results and confirm whether Trcv can be trans-airway transmitted. So first we look at the whitefly airway and oocyte development, because trans-airway transmission must wear the airway. So this is the newly emerged whitefly, and it shows the airway is still unmature. And this is 2 days after emergence, 6 days after emergence, and 11 days after emergence. It shows the airway become mature gradually, and we call this phase 1, phase 2, phase 3, and phase 4. We look at the localization of Trcv in whitefly airways. Interestingly, we found when we put the whitefly at a different development stage on an infected plant, only the virus can only be detected in the mature whitefly, such as 6 days or 11 days. So this green signal is the virus. So it suggests the Trcv can infect the airways, but it can only infect the mature airways. So we did another experiment to check Trcv trans-airway transmission. So we put the whitefly on Trcv infected tomato plant. Then after virus acquisition, we transfer the whitefly on a cotton plant, which is a no-host plant of Trcv. We let the whitefly to lay eggs, and then we remove the whitefly and wait for the eggs to be developed. So all the viruses actually are from the eggs. We collected eggs, nymphs, and adults, and then checked the percentage of the samples with virus infection. So this is a very interesting study. This result shows. So if we use 11 days whiteflies after emergence, it can transmit the virus to the next generation eggs, nymphs, and adults with very high efficiency. And it can also transmit the virus to a new plant. However, if we use a young mother, it's just a newly emerged whitefly to do this experiment, it can show this can very poorly transmit the virus to the next generation, and it cannot transmit the virus to a new plant. So it suggests the Trcv can be hourly transmitted, but only by mature whiteflies. So the vitellogenin VJ play an important role in hourly development. So we checked the expression of vitellogenin at a different development stage. We found the VJ expression gradually increased and peaked at the 11 days, which is coincident with the presence of virus hourly. So we guess maybe the VJ is involved in virus hourly transmission. Then we checked the co-localization of Trcv and the VJ by immunofluorescence. So in phase 1, there is no VJ, and therefore no virus in the airway. In phase 2, when the VJ is present, the Trcv also present, and they nicely co-localized together. And also in phase 3, this picture also shows the co-localization of Trcv and the VJ at different magnification. Next, we examined whether other bigmavirus can be trans-hourly transmitted. We used the papaya leaf China virus, which is another bigmavirus affecting papaya. We put the whitefly on papaya leaf China virus infected tomato plant, and then after virus acquisition, we transfer the whitefly on cotton plant, and we let it to produce eggs. Then we collected eggs and nymphs and adults. This figure shows both bee and kill whiteflies, and both young and elder whiteflies. No papaya leaf China virus can be transmitted to the next generation. So it suggests this virus cannot be trans-hourly transmitted. So what's the reason? So we checked the infection of this virus to different tissues of the whitefly. In midgut, this virus can be found in the midgut and in the cereal gland, but it cannot be detected in the airway. So it suggests this virus cannot enter the airway. As I have mentioned, the VJ is important for the interaction and the enter into our airway. So we checked the interaction. We found the whitefly VJ can only interact with Trcv CP, but not papaya leaf China virus coat protein. Because the CP coat protein is the only determinant of the vertical transmission. We also exchanged the coat protein of Trcv and this papaya leaf coat China virus and generated 2 mutant viruses. All those viruses can infect plants very effectively. However, when we look at the trans-hourly transmission of the mutant viruses, now we found the mutant Trcv. It can be found in mid-gut, in primary cell gland, but it cannot be found in the airway. And for another viruses, it can be found in all the tissues. So it suggests the coat protein is important to determine the trans-airway transmission of this virus. And we also checked the trans-airway transmission of mutant viruses, and it's consistent with previous study, and the mutant papaya leaf coat China virus now acquired the trans-airway transmission ability. We also silenced the beta-longinin, and we found that when we silenced this protein, it cannot affect the acquisition of the virus in the whole body, but it significantly decreases the amount of virus in the airway and the trans-airway transmission. So in summary, this result shows the Trcv trans-airway transmission is dependent on wet fly airway development. The wet fly VG can mediate the trans-airway transmission of Trcv, and this vertical transmission can contribute to its global invasion. So except the transmission of the virus, we also noticed several interesting phenomena in our lab. We found when the healthy wet fly, the no virus wet fly always prefer to feeding on infected plant. And when it gets the virus, some of the viruliferous wet flies will choose to feed on healthy plant. So it means there are complex interactions among plant, virus, and wet flies. So our first question is, will virus infection of host plant affect plant defense against the wet flies? Therefore, the wet fly prefer to feeding on the virus infected plant. So we did a very simple experiment. We put wet flies on the healthy plant and on the virus infected plant and checked the survival rate and fecundity of the virus. We found the survival rate and fecundity of wet flies on Trcv infected plant were much higher than those on uninfected plants. So which gene, as I have mentioned before, the Trcv only encode 6 proteins. So maybe one of the proteins can suppress the plant defense to wet flies. So previously, it has shown the C1 and C3 proteins. C1, this is C1, and C3, they are involved in virus replication. And the V1 is the coat protein. So they are not possible to involve in, to manipulate the plant defense. So we generated 3 transgenic plant, C2 transgenic plant, C4 transgenic plant, and V2 transgenic plant. We found only the transgenic C2 can increase wet fly performance. It means the C2 is regulating plant defense to wet fly. So how Trcv affect plant defense? We collected healthy plant with whiteflies, virus infected plant, and virus infected plant with whiteflies, and did RNA sequencing. We found that the transcription of genes in jasmonic pathway were significantly decreased in virus and whitefly co-infected plant. It suggests that because the jasmonic pathway, JA pathway, has been reported to participate in anti-insects. So it suggests the virus infection can suppress plant jasmonic acid pathway. They also checked whether the C2 transgenic plant can affect the jasmonic pathway. Indeed, we found the transcription of genes in JA pathway were significantly decreased in C2 transgenic plant. This means the C2 can suppress plant JA signaling pathway. So how C2 regulate plant JA signaling pathway? They did East2 hybrid with C2 as a bait, and we found several proteins interact with the C2. And one of the proteins called ubiquitin 40S ribosomal protein S27A got our attention because this protein is maybe involved in the degradation of JADs, which is a negative regulator of JA signal pathway. So it confirms the interaction by East2 hybrid and JST pull down between C2 and RPS-27. So next, we want to confirm whether RPS-27A play an important role in tomato defense against whiteflies. When we sequenced these genes, we found the survival and fecundity of whiteflies were significantly higher on RPS-27 silenced plant. So it suggests this gene really plays an important role in plant anti-whitefly. We also found silencing of RPS-27 can suppress the expression of terpenes in synthesis gene and the degradation of JAS1. And the C2 can also interact with RPS-27 to suppress the degradation of JAS1. So based on those results, we proposed a model. So in the healthy plant, when whitefly feeding on the plant, it can activate the J signaling pathway by the degradation of JAS1 and cause the production of downstream defense genes. However, when the whitefly feeding on a virus-infected plant, the virus C2 can block the degradation of JAS1. Therefore, it can block the downstream expression of defense genes. Therefore, it's beneficial to the whitefly survival. So just now, we have mentioned that the virus infection can suppress plant defense to whitefly. So we want to ask another question, whether whitefly feeding on plants will affect plant resistant to viruses. It also did a very simple experiment. So we have control plant and the plant with the whitefly pre-infestation. Then we use virus to transmit, use whitefly to transmit the virus on those two plants. Then we detected the amount and the virus infection rate in the plant. We found on the whitefly pre-infested plant, the virus infection rate and the amount of virus is significantly lower compared to the control plant. It suggests the wet fly pre-infestation can spread the later virus infection. However, people may wondering, the wet fly pre-infestation may affect subsequent wet fly feeding. So this kind of expression is probably because it affects wet fly feeding. So to test these possibilities, we did another experiment. We have the control plant and the wet fly pre-infested plant. Then we did agro inoculation, means we can use agro bacteria to infect the plant. It can exclude the effect on wet flies. Our result shows the quantity of Trcv genomic DNA also decreased in wet fly infested tomato plant, even by agro inoculation. So why wet fly inoculation can affect the later virus infection. So we take the control plant and wet fly pre-infested plant and did RNA-seq. We found the wet fly pre-infestation activated the plant salicylic acid signal pathway very effectively. So it's maybe the salicylic acid pathway causes the plant resistant to virus infection. Then, as we know, the wet fly feeding can activate the acid signal pathway, but the cotton bollworm feeding can activate the GA signal pathway. So we checked whether the cold bollworm feeding can affect virus infection. So we had the control plant and the cotton bollworm pre-infested plant, and then we using wet fly to do virus transmission. Interestingly, we found the quantity of Trcv genome DNA also decreased in tomato plant under cotton bollworm infestation. And they found the cotton bollworm feeding can activate the GA signal pathway very effectively, but has no major effect on the acid signal pathway. And we found a lower portion of wet flies settle on the plant with the cotton bollworm. So it suggests the cotton bollworm feeding can activate the GA pathway, therefore suppress the wet fly feeding. We also examined the effect of SA and GA on Trcv infection. We found the SA treatment significantly repressed, where GA treatment promoted the Trcv infection. We also checked whether wet fly infestation and the SA treatment can affect the callus accumulation, because the callus accumulation will affect virus infection. So we found that indeed the wet fly infestation and SA treatment can increase the callus accumulation, therefore decrease the virus infection. So in the last two study, we found the virus infection of plant can suppress plant resistant to wet fly, and the wet fly feeding on wet fly can also affect virus infection. So our next question is, where virus infection of wet fly affects its feeding on host plant? So because wet fly is a phloem feeding sucking insect, during its feeding, this is its stylet. During its feeding, it can secrete many effectors into the plant and regulate the plant defense. So we would like to know whether virus infection will affect whitefly salivary effectors. So we take some healthy plant and virus infected plant and put on the whitefly, and after several days, we collected the whitefly, dissected the cellular gland, and checked expression of some effector genes. Interestingly, we found one effector gene, BT56, is highly induced in under virus infection. We also examined whether whitefly cellular gland BT56 will affect whitefly performance on host plant. We found when we overexpression of this gene on plant, the virus survival rate and the fecundity is significantly increased. However, when we silence these genes, it can decrease whitefly survival rate and the fecundity. We also checked the localization of this gene. We found this gene is only expressed in whitefly cellular gland, and it's indicated that BT56 is a saliva protein specifically expressed in whitefly cellular gland. Then we used the electric penetration graph to study the effect of BT56 on whitefly feeding. We found when we silencing this BT56, it can significantly affect whitefly feeding. We also looked at the mechanism of BT56 on whitefly feeding. We found when we overexpress BT56, it can significantly reduce salicylic acid production in whiteflies in the plant, but it has no effect on GA production. And when we silencing the whitefly BT56, we found much less SA was produced. It suggests the whitefly BT56 plays an important role in the activation of the tobacco SA signaling pathway. Then we also looked at for the proteins interact with BT56 from the plant by ES2 hybrid. We found a tobacco class 2 knotted one like a homeobox transcription factor can interact with BT56. And this gene is specifically expressed in the phloem cell. This is the phloem tissue of the plant, and they found this gene is expressed in the phloem cells. And BT56 is secreted into the phloem, so they may interact in the phloem and regulate plant defense. So when we silencing this gene, it can increase whitefly survival and the fecundity, and also this NTH202 can suppress whitefly-induced SA accumulation. So those results suggest BT56 can directly interact with NTH202 transcription factor and increase whitefly-induced SA accumulation. So based on those results, we propose that the whitefly infection can promote the secretion of BT66. Then the BT56 can bind to 202 and induce acid production, which can suppress the plant GA defense. Therefore, is beneficial to the whitefly survival. So in summary, in this part, we found that TRCVC2 can interact with plant RPS27 to suppress GA defense, therefore beneficial to whitefly survival. On the other hand, the whitefly feeding on plant can activate plant SA pathway and suppress TRCV infection. Finally, TRCV infection of whitefly can increase the secretion of BT56, therefore induce plant SA signal pathway and is beneficial to whitefly feeding. Okay, I think that's all for my presentation. I would like to thank my collaborators, Professor Shuzhen Liu and Professor Ping Zhou, and all the students who has working very hard on those projects. Okay, so thank you. Thank you for your listening. And any questions?
Thank you, Dr. Wai Weng from China. Any question, please? في حد كان رافع ايده يا جماعة عايز يسأل.
اتفضلي دكتور ومي.
So do you know how to remove this bar? It's really bothering me.
Thanks a lot for this great presentation and detailed explanation, especially biganovirus, plant viral disease by person sucking insects. I'd like to ask you about, from your point of view, what are the recent trends in control bemizetabaceae besides
Sorry, I cannot hear you very clearly.
From your point of view, what are the recent trends in control the whitefly besides this method? I'd like to say a chemical substance can inject into the plant and block the whitefly feeding.
You mean how to control the whitefly based on my study?
Sorry?
So
Yeah,
I cannot
yeah.
understand the question. You mean how to control the whitefly, right?
I'd like to contact with you and call you on private, please. Can I have your email address?
Okay, okay, you can contact me in private.
في سؤال تاني؟ في دكتور عبد العزيز عقيل بيسأل كم المدة التي تحمل الحشرة المرض حتى تموت وكيف يمكن الحد من انتشار الفيروس؟ كمل معلش ما سمعتكش. اوكي
اتفضل دكتور عبد العزيز.
So any more question? Maybe you can.
ماشي. منصور ماشي.
يا دكتور عندي سؤال بعد إذن حضرتك.
اتفضل.
الفترة اللي الحشرة بتقعد فيها بتحمل الفيروس ده عشان تموت قد إيه؟ وإزاي إن أنا أحد من انتشار الفيروس ده أو مكافحة الحشرة نفسها؟
This is me, Mansour, the interpreter. I will interpret the question now. He asking about to what extent this white fly carry disease, and after that will die, and how can I provide or control this virus spread?
الصوت واضح؟
So maybe we can find some key receptors for virus transmission. And with the development of CRISPR-Cas9 technology, we can try to mutate those receptors, and maybe it can help to stop the virus transmission.
Also, there is a
yes,
question.
I also see another question.
You can read it on the chat.
Yeah. Is there any further transcriptome analysis to detect gene expression following infection to detect susceptibility? Yes, we actually have done previously. We have done this transcriptome analysis with whole whiteflies. So now we are doing the tissue-specific analysis. For example, what happens in the cellular gland after virus infection? What happens in the midgut after virus infection? And we also look at the plant side. So what happens to the phloem cells of the plant after virus infection? I think that because now with the development of the single cell sequencing technology, more can be done. Yeah, I think there is another question from Hala Akada. The question is, what about spraying plants with some resistant induced substance as jasmonic acid, salicylic acid, aspirin? I think it's a very good suggestion. Very good question. Because we found the GA, the jasmonic acid actually pathway is very important in anti-whitefly. However, the whitefly is very smart. It does not induce the jasmonic acid pathway. So we can manipulate the plant, try to induce the jasmonic acid to let the plant become more resistant to the whitefly. Yeah, I think that's my understanding. I think it's very possible by manipulate the plant defense, we can have a plant is hair resistant to whiteflies. And the next question is, did you detect specific genes responsible for the resistance? Yes, actually, we also studied the whitefly plant interaction. I didn't talk about it today very much. We found that the whitefly feeding can really induce several unknown plant defense genes. Now we are working on the function of those genes, of those plant genes in insect resistance, and also because we found many plants, the whitefly is can feed on so many different plants, but we found for some relatives, the wild relatives of tomato, there are several wild tomato. It has highly resistant to whitefly. They think we can try to identify some new genes, resistant genes from those resistant plants. So my email is this is my email. So if you have any questions, you can ask me.
Okay, thank you, Professor Wai-Wen from China. Thank you.
Okay, thank you. So bye-bye. I think it's time for the next speaker. Bye-bye.
Bye-bye. Dr. Ashtosh, are you here? Yes. Okay, Dr. Ashtosh من الهند.
Yes, I'm there.
هيتكلم عن لمحة عامة عن دور النباتات الطبية في وقاية النباتات على الآفات.
My screen is visible to all. Can you hear me?
Yes. Yes, please, you can start.
Okay. Okay. Today, very good evening to one and all. Today, I'm here to present my topic that is quite relevant with this, not with the normal plants, but it is more of an care of a normal plant with along with the medicinal plants, the herbs, because like we use plants extract as to treat the disease or to for the curing remedies. As we know, we all have seen the pandemic of COVID-19, and especially in India and Southern Asia, they have boiled the stem and the bark of particular plants, which has the immunomodulatory effect further, which has given. So plant protection, just not using the plants as a medicine was an important topic, whereas taking care of these herbs and plants were an important topic as well. So these are some of the important areas where we need to. Let us see what our traditional medicine had to say about it. In Indian science, we say this tradition as Ayurveda. So what is Ayurveda is? Ayurveda has two Sanskrit words. One is Ayu, another one is Veda. That is, Ayu refers to the science of life. So what we say that the preservation of the medicinal plant leads from the Ayurveda, and there was a particular branch dedicated for treating the plants and plant protection that is called as Vriksha Ayurveda. The Vriksha refers to the trees and Ayurveda refers to the science of life. So Vriksha Ayurveda was an entirely dedicated branch in the ancient era, which were taking care of management of plant protection. So let us see what was there and how can it be related to today's era, and do we have some organic benefits out of that? So we will see all these particular things. So if we see this traditional knowledge is a vital for the sustainability of natural resources, including the medicine plants. Biodiversity conservation can be learned from the context of specific local knowledge and integral transmission of the knowledge, skills, strategy, concern for the well-being of the future generation. Now, coming to this Vriksha Ayurveda, which deals with the various tree and plant species, ensure healthy growth, productivity. The ancient text consists of about 170 different plant species, including herb, shrub, trees, and the different chapters of the deals with the agri, horticulture, home gardening, intercropping and storage, etc. Now, why this paper has an interest to understand the traditional knowledge on biodiversity, particularly in the light of contemporary research on traditional and the formal knowledge system, and demonstrate the value of a traditional knowledge for the biodiversity of conservation. Now, what this tradition refers to, we always state tradition, let it be Egyptian tradition, European or Indian. So what we take it as a contextual meaning, the religious tradition, that is like temples, forest, monastery, forest, sanctified, defied trees. Now, traditional tribal tradition, sacred forest, sacred groves, sacred trees, the royal tradition, the royal hunting reserve, elephant forest, royal garden, etc. Livelihood tradition refers to the forest, grove, serving as a culture and social space and the source of live-like products and services. For instance, the sacred groups were dedicated to the deity or a village god, protected worship like a Devakardu in Shimoga in India. This is the name of the place. So they are like for the sacred group are important in the diversity of the floral and faunal diversity that have been conserved by the local communities is sustainable manner. They are present in Himachal Pradesh, Maharashtra, Kerala, Karnataka, and other places, highlighting the community managed of the conservation of forest. The practice of allocating the trees specific individual were based on the lunar asterism, that is Nakshatravana, Navagraha Viksha, and Nakshatravana, Rashivana, and Navagraha Viksha. So they were based on our solar system. They used to classify the various plants. Like we see this, as we see this plant name in this is a Sanskrit name, this is botanical name, this is a star, and where it has the lunar and the solar plexus where they have a relation. For example, I'll not go into the depth of it, but for an example, like Butia monosperma, it has in solar system, it has the relation with the moon, and in the lunar system, it has the relation with the Chandra, that is the moon. So if we see the therapeutic use of the flower and harvesting system, seasons were also mentioned. Like if you have to use the latex, that it was early winter, harvesting in general was early winter, and roots and fruits like that, the very specific time were mentioned. Now, recognizing the significance of the plant bioresources and floral values in ancient India was emphasizing was has been laid on to the mainly the late conversation of the flora. Ancient texts contain the many description of the use, management of the forest, and highlight the sustainability as an implicate theme. The treatise called as Visha mentions about the plants, its importance in disease suffered by them, treatment, protection from the external factor, increasing the yield. Conservation techniques like protection of the plant from the mist and pests, etc. The chemical fertilizers show the dramatic short-term benefits, but in longer run, they are adversely impact the soil, water, perhaps nutritional quality of the plant. Hence, there is a great scope of integrating the traditional practice for the better productivity of quality of planting the material. The second chapter, which is Bijotpatavidhi, illustrates the process of the seed germination. The further explains about the grading and preservation of the seeds. Method described for the preservation is to mix the seeds with ashes, and it was also suggested that wheat should be exposed to the medicated smoke, which can serve as an antimicrobial agent. The fertilizers are prescribed for the undeveloped and underdeveloped trees and plants. Now, Dhruva Rakshasa is a chapter which deals with the several advices to save the plants and trees, weathers, and other conditions like windstorms. It also tells about the medicine plants, broken branch to protect them, the whole trees from drying. The use of the powders of Solanum indicum, Sesamum indicum, Ambalis rice, Brassica jucinea, milk, ghee, cow dung has been mentioned in almost all the texts for protection of the during the sources. So, in addition to the pre-treatment applicable to all the seeds in journal, treatment specific to the specific plant also have described. Various seeds in priming process have been carefully designed in the Vriksha Ayurveda to allow the early germination to obtain the good quality of seedlings by following the classical technique. A study conducted to compare the effect of the Vriksha Ayurveda and modern cultivation techniques on germination of Bakuchi has revalidated and germination behavior of the dormant seeds of the Tolare qualifolia. The chapter Chitrikarna describes around some astronomy techniques such as to make the plants bloom throughout the year, irrespective of the season, bringing forth premature maturity to the plants, fruits, changing the shape from the tree for nourishment of the plant. Use of a biofertilizer called as Punapajala has been mentioned. Now, what is this Punapajala? This is called as organic. This is something which is organic in nature. So, Punapajala is a natural organic product derived from the animal and plant products containing a significant quantity, one or more of the primary nutrients like nitrogen, phosphorus, potassium, which are necessary for the plant growth. The literary meaning of the Sanskrit word punap is smelling like a dead or stinking. The name apart is for the liquid manure which is prepared using the excreta, bones, body, flesh, marrow of the animals, fish, decay, plant products, etc. The Punapajala has some plants growth regulatory action through which it enhances the overall growth of the plant. Being a liquid fertilizer, it is more suitable for the manure and can be beneficial in growth of medicinal plant with probably minimal toxic effect of the human body when compared to the chemical fertilizer. Usually the raw organic matter decomposed into the humus, which will be further digested by the soil microbes, reducing the high level of the organic acid like humic, carbonic, fulvic acid, increases the high tension exchange capacity. This capacity is responsible for the mobilization of calcium, potassium, and other plant nutrients. In order to obtain a good result, aerobic composting is said to be beneficial. The nitrogen, which is very essential for the plant growth, is supplemented by the blood, cotton seed, fish meat, emulsion, whereas compost from the bird manures, bone meal are rich sources of the phosphorus, potassium, which helps in regulating root, bud, flower, fruit formation, cell division, sugar formation in sap, chlorophyll production, photosynthesis, increasing crop resistance to the disease, etc. The other important micronutrients are magnesium, calcium, zinc, manganese, copper, iron, and selenium, which are also supplemented by the organic compost called as Punapajala. Researchers suggest that application of the principle of Vriksha Ayurveda like Punapajala does produce the phenomenal and interesting results. Since few research work have been carried out, this discipline of science needs to be developed through concerted research effort to ascertain its utility. Now, what are the advantages of this organic farming? Though chemical fertilizer increase the yield, they pose a certain serious health threat to the human being, especially infants, pregnant, and nursing mothers. Specifically, these are the main area targeted when the herbs and the plants used in the medicine part. Especially when we talk about the specific plants like Phenospora quadrifolia, the main cucurmin, and other plants which are used in day-to-day activities for the food items plays an important role over the human immunity. Another concern driver of the health is the contamination of the medicine plant with the toxic heavy metals like mercury, lead, cadmium, through fertilizer, harmful industrial waste contaminating the water sources. In contrast, organic manures are considered to be safe, yielding good produce by improving water penetration, biomass, nutrient availability, growth, heat stress resistant, and it also helps in improving the soil pH, which it has as impact on the plant growth. And the soil studies have using the Punapajala for growing senna have shown the total phenoside content per plant for Similarly, Langley, the Gloriosia superba, the active principle methonic extract was found higher in amount. When Kunabajal was used for brinjal, it produces large number of branches, high yield, fruits with the lesser seed, lower susceptibility to the disease when compared to the plants grown with artificial fertilizer. Similarly, for the mango, coconut, chili, paddy, vegetables, etc., similar results have found. Thus, Kunabajala is by virtue of its behavior as a growth regulator has been effective in increasing the leaf area, higher yield, flower, fruit as well, photoconstituents. Some major centers carrying out the Vriksha Ayurveda related work is CIKS, Chennai, Asian Agri-History Foundation, Secunderabad, that is again in India, and National Institute of Vriksha Ayurveda, Jhansi. Professor Nene and his group at AAHF promoting the Vriksha Ayurveda in big way, whereas CIKS Chennai is involved in promoting the organic farming, works along with the farmers belonging to the various villages in Tamil Nadu. They are also involved in testing and validation of the indigenous knowledge of agriculture by rapid assessment of the traditional agricultural practice. As a result, these experiments, that of Indian Council of Agricultural Research, using the modern research procedure, it has proved that traditional knowledge is valid beyond the doubt. To conclude, I must say the use of the Panchagavya, that is, the 5 products which are produced by the cow milk, the cow dung, cow urine, the curd which is returned out from the milk, and the urine. These are the main products. Other mentioned in the various tests can be studied further for the efficacy, if found it to be sustainable, can be adopted for the various steps involved, developed for the organic nursery protocol for the medicinal plant. When we talk about the cow should be indigenous variety because the specific type of protein, the A2 protein, is present in the indigenous cows. The majority of the raw materials used in this procedure are byproducts obtained from the other activities are easily available around us. The procedures are easy and economical too, which is added advantage. Many of the raw materials like listed in text of Vriksha Ayurveda, such as flesh, bone of animal, husk, oil, cakes, dung, urine of cattle, etc., are waste products, and utilization and recycling of these products will also result in effective waste management. With help of the Asian text, modern methods of agriculture, we can not only scientifically prove the saying of the text, but could also establish some novel modified methods for agriculture system. The proper interpretation and availability of Vriksha Ayurveda can also play an important role in the field of intercropping. Putting forward for the use of the organic fertilizer can play a crucial role to build an eco-friendly environment, and attempt has been made to compile the traditional method for conservation and prevention of the medicine plant. It is hoped that the Asian wisdom coupled with the modern technology would benefit the mankind. When we talk about the uses of the plant, the Acharya Shushruta, which is also, he was also recognized as a father of surgery in many medical schools. He has coined the term Sarvam Dravyam Panchabhautikatvam. That means the 5 factors which compounds which contains by all. Any plant can be used for the therapeutic personal purpose to treat the mankind and help this. Thus, this part of protection and taking care of the plant is of more importance, how it states its utility in difficult time of the corona and COVID-19, where Tinospora cordifolia was effectively used for making the decoction and was used as an immunomodulator, and which has decreased the rate of mortality in Indian country. So with this, I want to conclude. These are my references. I would like to thank organizers to give me an opportunity to talk on the topic. Thank you one and all. Any queries? Any queries anybody has?
Any question? في أي أسئلة يا
في
جماعة؟
صندوق الأسئلة في حبة أسئلة، في مجموعة أسئلة في
طب
صندوق
أوكي، افتح لهم من فضلك يا باشمهندس. فتحت لهم باب الأسئلة؟ مهندس سعيد.
في مجموعة أسئلة في صندوق الأسئلة. دكتور صلاة طالب سؤال.
طب خليه يتفضل.
I once again thanks organizer. I guess there are no queries. I once again request the audience that to use Vriksha Ayurveda's references in current era for the plant protections, and to use and prepare this organic manure and do the extensive research in not only medicinal plants, in all plant cares. I hope it would be beneficial to the audience, what I said, and extensive work will be carried out as I'm also doing on the Panchagavya, and it will benefit the organ. Once again, I thank organizers. I would like to take my leave by saying this, and thank you one and all, and we'll meet once again. Okay, bye-bye.
Okay, thank you, Dr. Ashutosh from India. Professor Dr. Layo, are you here?
دكتور لايو، هل أنت معنا؟
بروفيسور دكتور لايو من الصين، الأكاديمية الصينية للعلوم الزراعية الاستوائية، هيقول لنا محاضرة عن رصد مكافحة الآفات عبر الحدود. دكتور لايو, can you start please?
Yes, it's my turn. Can you hear me?
Yes, please.
Okay, thank you, Chairman Dr. Noha. It's my honor to give a talk here. Okay, I share my screen. Where it is. Okay, now let's start. First, I would like to introduce myself. My name is Li Baoqian. I come from Environmental Plant Protection Research Institute, Chinese Academy of Tropical Agriculture Science. Today, my presentation is about monitoring and control cross-border insects, actually mainly in Hainan province. Okay, for your idea, I'll give the briefly give the information on Hainan. Hainan actually is a tropical island. Not big, not large island. It's this is the area, and also the population is about 9 million, and the temperature is high. Because it's a tropical island, so the pest can breed all year round. And I need to point out, Hainan is a free trade port. So that means there are more trading, and more trading means more invasive pests. And also, Hainan is also a seed breeding base because it's a tropical area. So in the winter, it also can ground the plants there. So that means that some pests can fly up and fly back for overwinter. So for the cross-border pest, mainly first is about the migratory insects, such as the four-armed worm that is now widespread in the world. And also some very common pests you may know, for example, is rice plant hopper, rice leaf ruler. So they can fly up north of China and back to Hainan. Okay, I just wanted to tell some research we have done about the pest four-armed worm. It's also invasive pest. Four-armed worm is Stoptra frugipta. It's native to tropical and subtropical in America. It's about 2013. It's first detected in Nigeria, in West Africa. And then this the map showed the spread situation in Africa. This about April 2017. This now it's about 1 year is almost the Africa. This now in 2018, you can see the map. That's green color. And this is the picture we took from Uganda and Liberia. That means it's widespread from Western Africa to Eastern Africa. And it's caused serious damage on the corn. And you know, the corn is the main food for lots of countries. So that's a big problem. As for Egypt, we went to Egypt in 2019, December 2019. When we test the information, the news from the internet, the pest is detected first in 2019 in up Egypt. And as Dr. Mao Sanada said, it happened in 2019, and now it's widespread in the corn crops and other crops, but not dangerous. During our trip in Egypt, we did not find any four-armed worm in the field of Nile Delta in 2019. That's the field trip picture when we're there. And for other countries, this is from in India. This is the India. So you can see this. The map shows how do the foreworm arm is spread all over the India country. That's the basic information you can see. And also in the Sri Lanka, it's spread very fast. In only 1 year, they can all over the country. Oh, some problem. Okay. This forearm worm, this in Burma, I think. Yeah, and there's this is covered here. Invaded in Burma in 2019 January. And also in Thailand, that's the map shows how spread in the Thailand. You can all over the country now. So here the slide is about the spread or worldwide. You can see all over the most of the country is now is find this pest. As for China, Hainan, we first detected the pest on April 13th in Haikou. And in China, we first detected country is in Kunming province. So after the invasion of the pest, we do the random survey in the field, not only on the corn, but also on the other crops, such as the sugarcane, because the pest also damage sugarcane, even other crops. So because Hainan is a tropical island, I mentioned before, so it can overwinter the pest. This during the 2019 winter, we do the survey in the field. So this the map shows the density of the pest. That means high density here, and but this also dodgy map because the density in the density of the pest in the field depend on they spread chemical or not. If they spread chemical, so you can you hardly find the pest. If they cannot, if they did not spread, you can find lots of pests in the field. Okay, this slide shows the different stage of the corn, the damage symptom. This seedling stage, this is trumpet stage, this the flowering stage. Even on the this the mature stage. So they also different the damage on different stage. Generally speak, the damage is serious in the early stage of the plant. And also we do the monitoring by the trapping pheromone trap. You can see this you can see the map, the dynamic. Usually we have 2 peaks of the pest in the later in the earlier November and also in the middle of the March during this 2 peaks in Hainan province, and also there are different dynamic situation on different spots. From our research, our calculator suggests the pest can have about 9 generations in Hainan. So this is the tropical area in China. as for the monitoring, there are different ways. First, we use the hormone trap, and also use the high altitude light. This light will can a very strong light, so can attract the moth more easily. And also we use the insect radar monitoring for monitoring. You can see we can catch the mouse use the pheromone trap, and this is different type of the pheromone trap. You can see that they also can they both can catch the mouse. This is we do the survey on the different sites to do to monitoring the pest, the city, the dynamics, so we can do the prediction. This we do also have the high altitude lab. So this is sure this can, and you can see this the light very high. When the moth fly over the light, they can be attracted on the trap. Okay, they show the different sites, the traps, the dynamics. You can see that this is different. Different sites have different models. So generally speaking, they have a couple of the peaks from the light dot. As for the radar monitoring, we have the different types of radar. This is a vehicle mobile insect radar. You can see this is the working situation here, and this map shows the frequency of the pest catched by the radar, and also can see the direction where they from. So we also do the genetic type. As you know, the four-armed worm have two types. One is rice type, one is a coin type. For our molecular detector testing, we found most of the moth is rice type. As for the controlling, we do lots of work on the chemical selection. From our work, they have a type of have many chemical can work very well. For example, this chemical and also this chemical do really good work. Because the density of the pest, there are lots of pests in the field during the growing season. If we do not controlling, that will no yield at all. This picture you can see, this is so different. This is controlling. This is after no treatment, no spraying, chemical spraying. You can see the coin is damaged all. And we also do the research on the UAV. UAV is a good method to control the pest because UAV can save a lot of labors. It's very effective. And usually, when you do the spraying, usually it's about night time, or because during the night time, the pest can come out. It's easily to kill them, and also the weather and also the humidity is good for the UAV operating. And we also do the trial on the seed dressing. Seed dressing is did some work, but not really work well as we think. So That means we need to do further research on that. We also do the research on the native parasitoids because there are lots of parasitoids. This native in Hainan province. There are 5 species parasitoids. You can see this for the egg parasitoids, and this for larval and also pupae parasitoids. As for the chemical, we also do the chemical, select the chemical, not only for controlling the pest. Also, we want to find the chemical safety to the parasitoids. We found that this the best chemical is this chemical the LC is really good, and also for safety, this chemical is best, we think. So we also do the field trial for the parasitoids. We releasing the parasitoids in field. It did work, but the situation is if there are lots of pests in the field, it's hard to controlling by the parasitoids. But if the low density of the pests, it work well. So because the pest is new for us, so we do the first we do the training, let the farmers know, recognize the pest, and also also teach them how to control the pest. Just we also do the batches to deliver to the farmers. Okay, the second part is about the invasive insects, because the first part is in cross-board, the pest cross-board by the fly. And the second part of the pest is invasive pest. It means invasive by the human activities. First is Brown-Tispa longissima. Here I introduce the pest. It's mainly about the palm pest. The first palm pest is serious damage in Hainan. Damage the palms in Hainan is Brown-Tispa longissima. Its common name is coconut leaf beetles. As you can see the picture, here they damage the palm. They usually damage the palm is the coconut. It can cause 90% coconut trees infested and about 40 to 80 loss in the coconut production in Hainan. It's first invaded in Hainan in 2003. Palm plant is imported. I think it's also important for Egypt, particularly the data palm. In Hainan or China, the palm is coconut, and also the betel nut. Betel nut is for tree for the locals, and also for the landscapes. So this the map shows currently distribution of this Brown-Tispa longissima. It's native native to Indonesia and Papua New Guinea, and now it's widespread in Southeast Asians. Well, something wrong with this slide, but doesn't matter. This, and we also do the research on the to show the inter-country trade of the palm plant is strong, had a strong relationship with the interception of the records of the pest. And this the genetic diversity of the pests across the Southeast Asian. This profiled sample similar to this from various countries in its invasive and native region. This means they can introduce by the loss of resources, not only from one country, maybe from Vietnam or from India or Thailand. And also this pest have very special biological traits. For example, they can burying the stuff. They can access. They can recover quickly after access the plants. That means they can overcome the negative environment. So we do the prediction using the climax, shows the map in the world. It means this pest will is suitable. This pest, most of the coconut countries will suit for this pest. So we must be alert this pest. And so this climate situation map for this map for the pest with particular reference to the world key coconut growth regions. The pest is hard to control because the tree is high, and also the pest hiding in the fruit, and also this high population growth. If we only use the chemical control, will cause the 3I problem. So we found the natural enemy is a good way to solve the problem. And we introduced 2 species parasitoids. First is the larval parasitoids, Acidification, and the pupae parasitoids. After we introduce this, we do the risk assessment. After permission from the government, we do releasing in the field. So here, the next I brief show you the process of the massive ring the parasitoids. So we first ring the host, means brown tispa, the beetles, the larvae for the larval parasitoids, the pupae for the pupae parasitoids. And we cut first at the beginning, we're using the central leaf, but the central leaf is hard to get, because if we cut the central leaf, will damage the tree. So we use the mature leaf together with the central leaf to rear the beetles. It's do good results. So we do the parasitoids in the room and the massive ring, and we also releasing in the field. We releasing in the field use the simple design. This design can keep from the rain and keep from the ants. Before we releasing the parasitoids, we do the survey, the population of the parasitoid, the pest. So this according to our research or study, when we do the releasing, that's ratio parasitoids and the host is 2:1, and also we also do the mixture with the 2 species and different containers. So you can see this is mummy by the attacked by the parasitoids and recovery of the trees. Actually, we controlling the beetles not only by the parasitoids. We using the IPM. IPM using the parasitoids, the fungicides, also use the pesticide bag. Pesticide bag like the tea bag. We insert the tea bags, this pesticide bag in the center of the leaf, as relying on the raining water, they can releasing the chemicals slowly and can controlling the pest for a long time. And for in medicine, we also do spraying chemicals. Okay, the second palm pest is the coconut blackhead caterpillar. This is also a moth. It's a moth. Its Latin name is Opsina original. This is the fourth stage of the pest. The moth, the eggs very small. It's hardly to find in the field, and the larvae can make the tunnel. And this is the pupae. Different from the Brontisma longisma, this moth damaged the tree from bottom to the top, while the beetles damage the tree from the central to the bottom. That's their difference. Different from the coconut leaf beetle, this moth can damage lots of the palm trees. For example, this is the king palm tree and other these palm trees. You can see this is the tunnel by the, made by the larva. And from our molecular research, it showed this all the moth is came from the India and then to Thailand, Malaysia, and then China. That's simply the root of the pest spread. It's native in India and then to Thailand and then China. And this prediction map for this pest now is the distribution of the moth, and this the prediction map. I think even in Egypt is possible. Can be is be aware of this pest in Egypt too. And also because we also found lots of the parasitoids, these different types of parasitoids. We also rearing these parasitoids, mass rearing in the lab and releasing in the field. And it's you can see the two pictures. It's very significant controlling results. This recovering trees. For the monitoring, we use the light trap. The light trap shows this the difference for the female and males. It's about 350 to 100 nm for the males and 368 am for the males. And this we do the light trap, and we also do the pheromone trap. So that's for monitoring. Okay, the third pest is called the red palm weevil. It's the wildest bird in the world, and I think also in the Egypt. This is the adult. These different type of these beetles, and this the egg is quite big, and this the larva and the pupae, and it also is the cocoon. So this the map showed this the distribution in China. It's actually it's widest spread in China. It's mostly distributed in the south of China. You can see the map here. It's damaged here. It's in the bone in the strong the inside of the tree. You can see the pest here. Serious damage. So for this, we also do the IPM method. First, we do the monitoring, and also do the biological or ecological research. This we use the diet for rearing in the laboratory, and if we first found this pest, we need to do the delete the trees. Is maybe the best way to do. For the monitoring, we use a visual or inspect tree by tree. You can see the damage in the field, and also you can see the hole in the tree, and maybe you can use the detector, doctor's detector to find this pest. And the best way, I think, is the pheromone trap is the best way to monitoring the pest. Here is the shows the best we do the research how high or how far to set the pheromone trap here, and also see the dynamic of the pest. Usually, is the peak is on the summertime. And we also select the chemicals. The chemicals not really work well, but some chemicals is do good work. For example, this chemical, and this chemical. Even this one is all do can control above the 17% of the pest. And then we also do the trial, use the chemical injection. And also use the fungicides, the BT. We do this is work on do some work too. The fourth pest is the renal beetle. Renal beetle, you can see the is renal here, so we call it renal beetle. Renal beetle is native to Southeast Asia, India. Now it's wild spread in India, Indonesia, Japan. Philippines, Sri Lanka, Vietnam, and it's also introduced to the Pacific Iceland. For example, in 2013, it's first discovered in Hawaii. That's the left tip of this pest. You can see the larval, and for the tree, you can see easily to tell. It have the V cuts. You can tell the damage by this pest. This is the picture took by in the ground in 2013. So that pest is sometimes for the farmers it's hard to tell this pest between this pest. This the picture shows the difference. And for the monitoring, we do use the pheromone trap in the field. As the result shows that it work well. For the chemical control, we do the research. Here is do the different trial, and I think we also use the nematode for the biological control. It do work. Okay, this is the fungicides, also the fungicides and the nematodes together to do the work. Okay. So at the end of my presentation, my recommendation for controlling the cross-border pest, I think cooperation is more important. And the first of all is monitoring. First, we do monitoring so we can do prediction and even do the risk assessment. So we know. We are aware of this pest and know this damage is dangerous, so we can put more attention on that. And also for different pests have different method. Generally speaking, RPM is important because the different pests have different biological and ecological. First, we do the research on the biological and the ecological, know how to control the pest. And my point of view is sharing the experience, sharing the information, and sharing technology for controlling the invasive pest is more important. Okay, and that's all my presentation here. Here is my information. I welcome you contact me if you have any question. That's all. Thank you.
Thank you, Professor Layo. Any question, please? دكتورة أنوار أباظة ليها سؤال، تتفضل.
افتحي الصوت عندك دكتورة أنوار، افتحي المايك.
دكتورة أنوار المايك عندك مقفول، افتحيه من فضلك. طب دكتورة مارجريت عدلي رزق، اتفضلي يا دكتورة مارجريت. المايك اتقفل برضو عندك يا دكتور مارجريت.
افتحي المايك عندك دكتورة مارجريت.
Sorry. كده open كده now. I wanted to ask how many trap used in the field for controlling for army war, and I wanted to ask about what is the time for using trichogramma and how many cartridge use.
Okay, for the trichogramma, from our research, it's not work really well. Usually, we releasing couples parasitoids together. I think it's earlier is better. So it's important for monitoring the pest in the field. When the trichogramma is attacked the eggs. If so, it's important for you to know when to releasing the trichogramma. Is it right?
دكتور مارجريت.
نعم.
سأل حضرتك ده اللي تقصديه؟
Yes. Thank you. About the trichogramma, but I did not
دكتور
tell
زينب
me about
دكتور زينب عواد. اتفضلي يا دكتور زينب. انتوا بتقفلوا المايك يا جماعة. طب دكتور أحمد المرسي. برضه قفلت.
الو السلام عليكم.
علي صوتك شويه يا دكتور.
الو سامعيني كده؟
ايوه اتفضل.
Thank you so much for valuable lecture. My question is, as a result of monitoring and prediction data, is there any way or the best way to avoid the infestation before it happens? Thank you.
Sorry, because the signal not really well. There's some noise there. Could you please type your question?
As a result of monitoring and prediction data, is there any way or the best way to avoid the infestation before it happens?
Could you please type your question?
What?
Could you please type, writing your question?
Okay.
Okay. Excuse me. Could I would like to answer the question about Ahmed. You said how to tell the difference between the red palm weevil and the renal beetles. I think it's easily from my picture I showed. For the renal beetle, it's damaged the leaves. It's have V cut. It's very easy to tell between the red palm weevil and the renal beetle. Though they both damage the trunk inside the trunk, but for the leaves, you can tell their difference. It's okay for you? For Ahmed, this question. Huh?
Yes. Yes, thank you.
دكتور محمد أحمد إبراهيم اتفضل يا دكتور.
Assalamu alaikum.
عليكم
Thank
السلام.
you, Dr. Leo. I want to ask about the head irrigation. You said to control red palm weevil.
Yeah.
So I need to know how to apply the head irrigation.
How what? The red palm weevil for controlling the red palm weevil, you mean?
About the use of head irrigation to control red palm weevil.
We usually use the trap. It's a farm trap. It's really good, work well, because usually the pest is hard to find. Even use the chemical spraying and the trunk injection not work well, but use the traps, you can kill the pest. The pheromone traps, I think is maybe the best way to control the red palm weevil.
Oh, okay. Thank you.
Thank you.
دكتور زينب عواد، افتحي المايك من فضلك واسألي. اوكي، في اي اسئلة تانية؟ تمام. Okay, thank you, Dr. Liu from China. Thank you, Doctor.
Thanks, Dr. Noha. Thank you. Bye-bye.
Bye-bye. Dr. Liu, are you here?
Yes, I'm here.
Yes. Welcome in Egypt. دكتور ليو، الصين الأكاديمية الصينية للعلوم الزراعية الاستوائية هتتكلم معانا عن مكافحة التلوث بالمعادن الثقيلة في الأراضي الزراعية. You can start.
Okay. Let me share my screen.
Okay.
Can you see my screen?
Yes.
So you can see my PPT right now? Oh no?
You can share your screen.
Let me try. Okay. Okay. Thank you, Dr. Noah, and good afternoon, everyone. I'm very happy to attend this workshop. It's my great honor to introduce you about my research. My topic is about the control of heavy metal pollution in intensive agriculture. I think it's a little bit more different with plant protection, but it's really have close relationship with plant growth and plant protection. For my presentation, there are 4 parts. The first is introduction. The second is challenges and objective. The third is the result and discussion. And at the end is some recommendations. For heavy metals, we all know that it's a kind of hazardous substance. They are included in the top of 20 hazardous substance of agency for toxic substance and disease Registry and the United States Environmental Protection Agency. And heavy metals like lead, cadmium, arsenic, and mercury are reported to cause several disorders in humans, including cancer, some disease, and the damage of our human bodies. What causes heavy metal pollutions? We know that heavy metals exist in soils, but how it can be pollution? From this graph, we can see the heavy metals emitted from traffic, factory, and mining. Well, if it had a large content, it will transport to a long distance and make soil contamination, water contamination, and food contamination. In the world, soil heavy metal pollution is a big issue. Soils are a main and terminal sink for heavy metal released into the environment by human beings' activities. From the table, we can see that globally more than 10 million sites of soil pollution have been reported, and with bigger than 15% of the sites contaminated with heavy metals and or metals. Heavy metal pollution has a combined worldwide economic impact estimated to be in excess of 10 billion US dollars per year. And in China, the soil heavy metal pollution is also an important pollution situation. From the graph, you can see about 19% of the agricultural soils are contaminated based on China's soil environmental quality limits, mainly with heavy metals and metals. They mostly come from fertilizer, manures, agrochemicals, and irrigation with wastewater, and also some solid wastes. Here, I want to talk about soil heavy metal pollution in intensive agriculture. That is because intensive farming can cause a hyperaccumulation of heavy metals in soils. It may from three parts. The first is the overuse of fertilizer like manure and pesticide. And the second is the use of sludge. The sludge may have a high level of chromium, nickel, lead, zinc, and so on. And the third is irrigated by wastewater, especially in some countries have the problems of water leak. Here we can see some examples about the soil heavy metal pollution in intensive agriculture. Agricultural practices such as application of fertilizers, organic composted and animal waste, and pesticides or herbicides have increased the amounts of many trace matters. For example, trace elements are found in animal manures since these trace elements are added to their diets as additives. Here are some examples. For example, in UK and in northeastern Spain regions, we all found that there is a high percentage of cadmium was due to manure application. And the second is long-term use of the other agrochemicals like pesticides, herbicides, and fungicides. These agrochemicals commonly contain like copper, zinc, arsenic, and lead. The most important concern is about copper. especially in some fruit grove like citrus grove in Florida, due to the repeated use of copper-containing fungi pesticides for sustained citrus production. Copper concentrations in some citrus grove soils in Florida have been found to be as high as 10 to 20 times greater than the background level. The challenges is that the problems of soil health introduced by intensive agricultural activities exacerbate the pollution risk of heavy metals in soils, as well as increase the difficulty of control of heavy metal pollutions. Why have these challenges? Then let's see. The first we focused on is soil acidification. Soil acidification is natural processes. Like in some varying regions, it has a high speed of soil acidifications. This picture shows soil acidification by vary in Hawaii. But the other reason introduced soil acidification is man-made processes, like the overuse of fertilizer to soils. From the table, we can see because of the overuse of fertilizers through a series of biochemical reaction in soils, the soil has tend to become more acidic. As soil pH declines, the supply of most plant nutrients decrease, where alumina and a few micronutrients become more soluble and toxic to plants. For example, alumina or manganese toxicity, cadmium or phosphorus deficiency, and also high risk of heavy metals. From the picture, you can see with the increased soil pH, the accumulation of cadmium in rice is increased. And the other soil problems induced by intensive agriculture is land degradation. It's induced by overuse and imbalanced fertilization, intensive speed slope agriculture, open mining, and other industrial waste. The result is it can make the soil have low fertility and low productivity, and also it will have a high risk of heavy metals. And the other kind of soil we focused on is the high water rate soil. In the subtropical and tropical regions, many soils are intensively water rate because of high temperatures and humidity. From the table, we can see the oxysoil and artisols are the main soil types in subtropical and tropical areas. Oxysoil has the character of acidic, low in phosphorus, nitrogen, and other nutrients, and limited in their ability to store nutrients, but have relative high soil organic matter content. And the artisoil also high in valuable minerals. It's higher than oxysoil, but also acidic and low in nutrients. For this kind of high water rate soil in general has a low content of organic matter and active clays. And these characteristics makes soils more prone to heavy metal contaminations. Moreover, this rate is often exacerbated by soil acidification, as we talked about before, resulted from fertilization. So the challenging is that we must to reduce the activity of heavy metals in soils and at the same time to improve soil quality. So in our research, we want to develop soil amendments which not only has a function to reduce heavy metals contamination in soil, but also can improve soil quality. We mainly focused on high weather rate acidic soil and acidic sandy soil. These results, at first we try to use warming compost. The warming compost is produced from warming composting manure under rubble trees. When we got warming compost, we used it in the heavy metal contaminated soils and to determine its absorption and soil characters, and also the immobilization effectiveness. These results, we got that the warming compost showed great absorption capacity for cadmium, nickel, and chromium, with a maximum absorption of about 13.3 microgram per kilogram, respectively. And for the extractable cadmium, nickel, and chromium in the contaminated soil, they were also significantly reduced by applying warming compost. We can see the lines in the lower in the graphs, and we also found that warming compost application increased the soil pH by 0.3 to 1.5 units, and there were negative correlation between extractable heavy metals and soil pH. So we also determined the soil properties, without warming compost and with warming compost. We can see warming compost enhanced the formulation of larger and water stable aggregates, and consequently decreased the mobility and availability of metals in contaminated soils. This study shows that warming compost is a potential option to reducing availability of heavy metals in high weather rate acidic soils. Then we developed this kind of soil amendments. We developed soil amendments with warming compost combined with oil and apatite, and to increase its immobilization effective. From the result from the experiment, we found that when we use the combined soil amendment, it also have a good effective performance. The available cadmium and the lead were decreased, and the combination treatment with I promoted the transformation of cadmium from extractable to iron and manganese oxide fractions, and also it promoted transformation of lead from extractable to iron and manganese oxide organic bites and residual fractions. And also the soil properties have been improved after remediation, with pH value improved by 0.17 to 1.33 units, and organic matter increased by about 13% to 124%. and the canyon exchange capacity also increased. So in summary, the combination of vermicompost and oil and apatite were more effective in immobilizing cadmium and lead in high weather rate acidic soils. And for the sandy soil, we developed a soil amendment based on phosphate rock. It is activated rock. This study, we compared the effectiveness of activated phosphate rock and biochar on the remediation of tropical sandy soils. We used the two soils, upsoil and spondsoil. This experiment is conducted in Florida. And we compared the three soil amendments, the dolomite phosphate rock and humic acid activated dolomite phosphate rock and biochar. From the experiment, we found that the phosphate rock and activated phosphate rock increased the absorption of cadmium and lead in soils, and the activated phosphate rock has most effective. And for the extractable heavy metals, it also be significantly decreased by amendments. And also the activated phosphate rock was more effective in reducing extractable heavy metals. For the port experiment, we can see that the heavy metal pollution inhibited by the heavy metal pollution inhibited plant growth, but the inhibition was decreased by amendments. And the concentrations of cadmium and lead in plants were decreased by using amendments. So in summary, the activation with humic acid increased the immobilization power of phosphate rock for cadmium and lead. And the immobilization of metals by activated phosphate rock is related to increased soil pH and available phosphorus. So at last, there are some little recommendations about the control of heavy metal pollution in intensive agricultural soils. At first, because of the resources, we should manage the fertilization and pesticides use. And for the contaminated soil, we can use soil amendments to reduce the activity of heavy metals in soil. And the soil amendment, the choice of soil amendments should, according to the soil characters. Here, according to our research, for the high weather rate acidic soils, we recommend vermicompost and its compounds. And for the sandy soil, we recommend the activated phosphate rock. This work is supported by major science and technology program of Hainan province, and also the foundations from University of Florida. Yeah, that's about my little research. I hope someone can well interested in it, and if you have the willing to cooperation, you can contact me. Thank you.
Thank you, Dr. Liu from China. في أسئلة يا جماعة؟ دكتور هدى سالم. اتفضلي يا دكتور هدى. دكتور هدى؟ طب دكتور سماح مصطفى. دكتور سماح. دكتور هدى سالم. Any question?
Maybe most of the researchers are not familiar with this.
Okay. Thank you, Dr. Leo.
Thank you.
بسم الله الرحمن الرحيم. تم بحمد الله الانتهاء من فعاليات ورشة العمل الدولية تحت عنوان دور وقاية النباتات في التنمية الزراعية المستدامة. وأتقدم بالشكر الجزيل للأستاذ الدكتور أحمد عبد المجيد مدير معهد بحوث وقاية النباتات لما قدمه من فكر وجهد ووقت لتنظيم وإنجاح الورشة هذه. كما أتقدم بالشكر للمحاضرين المصريين والمحاضرين من دول ألمانيا، الولايات المتحدة الأمريكية، الهند، الصين، والشكر لزملائي المشاركين في ورشة العمل. كما أتقدم بالشكر للزملاء القائمين على تنظيم هذه الورشة. والآن أقدم لسيادتكم أستاذ الدكتور أحمد عبد المجيد مدير معهد بحوث وقاية النباتات لإلقاء توصيات فعاليات ورشة العمل. فليتفضل سيادته.
شكرا. الصوت هو نوع سامعني؟
أيوة سامع حضرتك، اتفضل يا دكتور.
بسم الله الرحمن الرحيم. في البداية أود أن أتوجه بالشكر لجميع السادة المحاضرين المتميزين والذين أسروا الورشة بمعلوماتهم القيمة. كما أشكر جميع السادة القائمين على تنظيم الورشة والسادة المترجمين. وأخيراً وليس آخراً أشكر السادة الزملاء الباحثين على مشاركتهم وتفاعلهم والتي كانت لها أكبر الأثر في نجاح هذه الورشة. السادة والسيدات الكرام، تضمنت فعاليات الورشة الدولية لمعهد بحوث وقاية النباتات والتي عقدت تحت عنوان دور وقاية النباتات في التنمية الزراعية المستدامة خلال الفترة من 21 ل 23 فبراير 2021 مناقشة وطرح بعض المواضيع الهامة التي تدعو إلى اتخاذ خطوات حاسمة وملزمة لمجابهة الممارسات الزراعية غير المستدامة وأزمات المناخ للانتقال إلى مرحلة جديدة قوامها الاستدامة ومواجهة التحديات والتعريف بالطرق الابتكارية الجديدة والآمنة لمكافحة الآفات وتحكيم الاستخدام غير المرشد للمبيدات الكيميائية مع أخذ في الاعتبار سهولة تطبيق تلك الطرق وانخفاض تكلفتها وزيادة الإنتاج مما يعزز ضمان الأمن الغذائي. وتوصي الورشة على بعض النقاط الهامة منها تطوير التكنولوجيا الزراعية لتقليل استخدام المبيدات واستهلاك المياه من خلال استخدام بشابير ووحدات تجزئة متطورة متعددة الطاقات، واستخدام الملقحات البديلة ومردودها الإيجابي على الزراعة والمزارع بما يعزز الأمن الغذائي. 3. الاهتمام بزيادة زراعة المحاصيل الزيتية والخضر والنباتات الطبية والعطرية التي تساعد على زيادة الأعداء الحيوية والحشرات النافعة والملقحات وتقليل الإصابة بالآفات. 4. المحافظة على التنوع البيولوجي في البيئة ضمن أطر إيكولوجية وأهم ركائزها هي حماية البيئة من الملوثات والتنوع البيولوجي مما يؤدي دورا مهما في تحقيق إنتاج زراعي مستدام من خلال تعزيز إنتاجية الحاصلات الزراعية والتنوع البيولوجي، وهذا يعد أمرا ضروريا لضمان الحصول على إنتاج غذائي أكثر وفرة واستقرارا، إذ أن المسطحات الخضراء التي تتميز بثراء مكوناتها وتنوعها تعزز من عمليات التلقيح بالإضافة إلى تحسين المقاومة البيولوجية للآفات والحشرات الضارة وبالتالي زيادة الإنتاجية الزراعية. 5. استخدام التكنولوجيا الناشئة في المكافحة البيولوجية للآفات وتطبيق رش الطفيليات والمفترسات باستخدام طائرات بدون طيار والتطبيقات العلمية الواعدة في هذا المجال، وتهيئة جميع الوسائل البيئية للمكافحة البيولوجية، ودور المكافحة الميكروبية باستخدام البكتيريا والفطريات الممرضة للحشرات، والتنوع النباتي بتحميل بعض النباتات الطبية والعطرية على المحاصيل والتي لها بالغ الأثر في خفض تعداد الآفات، ومردودها الاقتصادي للمزارع عوضا عن الزراعة للنبات الأحادي في الرقعة الزراعية، مما يؤدي إلى زيادة العائد الاقتصادي للمزارع. سادساً، استخدام العديد من البدائل الآمنة للتقليل من استخدام المبيدات، ومنها أيضاً النباتات الطبية والعطرية والأشعة فوق المستخلصات الخاصة بالطبية والعطرية والأشعة فوق البنفسجية وغيرها من البدائل الآمنة. أيضاً هذا كله ليس عوضاً عن استخدام الطرق الميكانيكية في مكافحة الآفات الناقلة للأمراض الفيروسية، مع العمل على منع ري بمياه المصارف إلا بعد معالجتها معالجة ثنائية على الأقل لتفادي التلوث بالمعادن الثقيلة في الأراضي الزراعية. رقم 9، تواصل الرصد والتقصي بهدف الاكتشاف المبكر للآفات عبر الحدود والبيئات المتتالية حول حركة الحشرة واتجاهها ومنع دخولها للبلاد. 10، زيادة معدلات التدريب على استخدام تكنولوجيا رصد الآفات الزراعية العابرة. 11، العمل على عمل وتنفيذ ندوات عن التنمية البشرية للمرأة الريفية لجعلها منتجة وتحسين من أوضاعها لتكون مؤهلة لتحقيق التنمية الزراعية المستدامة. العمل على حماية البيئة المصرية من التلوث من خلال استغلال مخلفات المحاصيل بما يخلق فرص عمل وصناعات للريف المصري. العمل على حماية الملقحات البديلة لزيادة إنتاجية المحاصيل عن طريق زيادة التلقيح الطبيعي، مع العمل على التوسع في صناعتي النحل والحرير من خلال الحصول على سلالات نحل ذات صفات وراثية ممتازة، وعمل برامج تربية وانتخاب للوصول إلى هجن مصرية من دودة الحرير التوتية. ويشكل تلوث البيئة والتربة تهديداً مقلقاً للإنتاج الزراعي وسلامة الغذاء وصحة الإنسان، وناجم عن استخدام المبيدات بصورة مكثفة. لذلك لابد من تطبيق الكميات المناسبة والموصى بها من الأسمدة والمبيدات، والتغيير من الممارسات الزراعية التي تؤدي إلى تراكم هذه المعادن الثقيلة، والبدء في خطوات واتخاذ خطوات حقيقية لتنقية التربة من إرث التلوث المتوارث عبر الأجيال. وبكده نكون انتهينا من التوصيات اللي خرجت بها الورشة العمل، وبذلك نختتم فعاليات الورشة ونلتقي إن شاء الله في فعاليات علمية أخرى بإذن الله، والله ولي التوفيق. والسلام عليكم ورحمة الله وبركاته.
ورحمة الله وبركاته. شكرا للأستاذ الدكتور أحمد عبد المجيد مدير معهد بحوث وقاية النباتات، ونشكركم ونتمنى لكم التوفيق والنجاح إن شاء الله، وإلى اللقاء في فعاليات أخرى لمعهد بحوث وقاية النباتات. السلام عليكم ورحمة الله وبركاته.